a lot of clients, whether it's ADC or not, they're in the FIH stage, they need to understand whether or not they have a product where they're going to invest tens of millions of dollars into it. So they need to know, is it efficacious? Is it manufacturable? Is it stable? What can we do with it? And that way they can get through their tox studies and their phase 1 and then understand if they have the money to invest to go further. So information that can be calculated theoretically, PIs, molecular weight, those can be done just based on the sequence. But any hands-on data you have, because as you know, proteins fold differently, and a theoretical PI and an observed PI can be different.
Welcome to the Smart Biotech Scientist. I'm David Brühlmann, your host, and today we're diving deep into the fascinating world of antibody-drug conjugates with Amanda Hoertz, who's a Vice President of Analytical and Formulation Sciences at KBI Biopharma. Amanda leads 300 analytical scientists across KBI's mammalian network, tackling some of the most complex challenges in biologics development. We'll explore her journey from chemistry PhD to an industry leader and discover what makes ADCs both incredibly powerful and uniquely challenging to develop. Let's dive in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Amanda, to the Smart Biotech Scientist. It's good to have you on today.
Hi, David. Thank you for having me. I appreciate it.
It's a pleasure. Amanda, share something that you believe about bioprocess development that most people disagree with.
Well, this question makes me nervous, but I'll shoot for it because I don't pretend to know what everyone else thinks. I think one thing that a lot of people believe is that you can adapt almost any biologic to a single platform. And I think that different classes of molecules and formats need to be considered and need special considerations. I think we can benefit from a lot of the historical data and the potential power of machine learning to reduce de novo data generation, but a lot of these determinations are empirical and have to be done for that specific molecule.
Yeah, that's an excellent point. There's definitely a lot of specificity for each molecule. Before we talk about ADCs, Amanda, let's talk about yourself, because you've built an impressive career leading 300 scientists across KBI's network. So tell us how you got started in chemistry / the pharmaceutical industry, biotech, and what were some interesting pit stops along the way?
Sure. I love science. I love the logical portion of it and how you can understand something and do an investigation and rule out factors. And it's actually, funnily enough, for my poor family, become part of my actual personal life. I think about things in a very analytical way, similar to an investigation we do at work. My initial love of chemistry actually came from when I was in high school. My chemistry teacher was actually a PhD from Berkeley, and which was obviously atypical for a high school chemistry teacher. And she had very, very high standard. She'd put us all at the board, and we'd be so afraid of being wrong in front of the whole class that it was my major focus to make sure that I understood those concepts the best, because that was my nightmare. In 7th grade, I also became a type 1 diabetic, and it was so impressive to me that this little bottle of clear liquid, when you had such a small injection of insulin, could make you feel dramatically better or worse. So those things together really kind of pushed me in the science direction. When I was in undergraduate and graduate school, I worked on antibiotic resistance and understanding how we can modify those genes to try to make novel analogs of currently approved and perhaps anything that's becoming antibiotic resistant to make those on a scalable, economic way. So that kind of pushed me towards industry. When I got to industry, what I loved about it was the fact that the work I did in academia was very conceptual, and I didn't really see it being applied in the short term. Whereas the work I do at KBI, it's going into clinics, it's being used now, and some of them fail, but I see it being realized. I see people getting the benefit. Especially as KBI has matured, we've had a number of products that we started out preclinical and now are commercial. We see the benefits. We see people taking these drugs, and it's not just a concept that 20, 30 years from now it might happen. It's actually happening. We know people are getting benefits. It's a great feeling.
Now, specifically, you're now working in analytical and formulation sciences. What excites you most about working in this very part of biotech today?
There are just so many different options coming forward for patients. We're on the cutting edge. We get to see all the different treatments. And as I said before, some of them fail, and that's a natural part of the industry. But we see how people are innovating their thought process, what different drugs can possibly be offered to the patients. And as we see them getting more and more specific, especially with something like ADCs, we see less side effects. We see patients getting improved options. And it's very exciting to be part of that and to see that being realized.
I'm very excited to talk about ADCs because it's a huge trend in our industry, no doubt about that. A lot of people are developing ADCs, but tell us why they are so powerful therapies before we dive into the nitty-gritty of ADC development.
Sure. I mean, there is a payload attached to the antibody, and that can be either non-cytotoxic or cytotoxic. Obviously, the more potent ones, harder to handle, but they are allowing us to take cytotoxic drugs that in general couldn't be administered to a patient, but target them specifically for the tumor. So by using this basically directing antibody, you're able to get those cytotoxic drugs directly to those tumors. And because we have less of the bystander effect, which is where cells that are nearby are also implicated in the cell death, it's more targeted. General chemotherapy that started out a long time ago would only target fastly dividing cells. So that would target other things that aren't part of the target and would result in things like hair loss and nerve damage. By being able to target these with ADCs, we're able to really just target the tumor. And we're able to have these ADCs and immunotherapies really be a very specific treatment for the cancer drug.
And what makes ADC development very challenging compared to traditional monoclonals?
The same thing that's a benefit is the same thing that makes it very challenging. So we have worked on and manufactured at KBI non-cytotoxic, many of them ADCs. Where it becomes really challenging is when you start working with the cytotoxic payloads. Those obviously in very small amounts are designed to kill cells. Therefore, there's a safety risk for the employees handling them for testing, handling them for manufacturing them. The biggest risk is obviously with the free payload. So analytically, we handle that very carefully. We only need that for really one assay, the free drug, where we have to run a standard curve of the free drug and then run the actual sample to see how much has released. But handling that free drug to be able to run that standard curve is the risk to our employees, any employee that would then service that instrument. Conversely, in the process development and manufacturing space, they would need to handle an even larger amount of that payload to be able to conjugate it to the actual product. And so those are the highest risks. So making sure that we have the right safety protocols so that we are not exposing our employees to any of these deleterious effects, which are by design for the cancer cells, but not by design, obviously, for general population.
Can you give us just a high-level view of how ADCs are actually produced? I mean, you have quite a puzzle. You have the antibody, you have the linker, you have the payload. What's the general sequence, especially for people who might not be familiar with ADC development?
A lot of these ADCs are developed before they come to KBI, but some of them that we're involved in, there's an antibody. It's typically one that's already well understood, and companies will modify the amino acid sequence to target the specific locations where the linkers can bind to then get that payload attached. So by modifying how the molecule is formed, you're able to target specific numbers of attachment sites and specific amounts of that cytotoxic payload. It is very important that we have the analytical tools to look at both the antibody, the free linker, and the free payload. Because the more stable that is, the better. Obviously, we want it to conjugate, and then we want none of the free linker to be showing up. The free linker tends not to be a problem. It's just a sign that we're actually losing some of the payload. The payload, obviously, is a problem if we have high amounts of free payload. So we develop analytical tools to monitor that over the course of stability under many stresses, and understand whether or not agitation stress, freeze-thaw stress, temperature stress, duration of any of those stresses, including acidic, basic, and oxidation stress, are influential in releasing, either damaging the linker or releasing that payload. And that's the function of our stability studies, to quantify the amount of that free payload that gets released. It's also obviously impactful if we don't have payload attached, the drug is not efficacious. So it's usually more the free payload that ends up being the problem, because in small amounts it's impactful, but we want to make sure that the drug we're delivering also has that payload attached.
Let's make this practical. Let's assume I'm a CEO of a biotech company and we are developing an ADC and we're coming to you, to KBI. What are the first critical questions your team would ask my company to set up our analytical strategy for success?
So one of the things that's great about KBI is we're a bespoke services. We offer what you need. So a lot of companies have a platform approach. You give us this amount of material, or we produce this amount of material, and we do these experiments. We'll take in the information you have. So if you were coming to me with an ADC that you want to develop, I would understand what information you have. What do you understand about the molecule? What can we leverage from historical data? And then set up, based on your timeline and your budget, what we can further understand to get you to the next stage. A lot of clients, whether it's ADC or not, they're in the FIH stage, they need to understand whether or not they have a product where they're going to invest tens of millions of dollars into it. So they need to know, is it efficacious? Is it manufacturable? Is it stable? What can we do with it? And that way they can get through their tox studies and their phase 1 and then understand if they have the money to invest to go further. So information that can be calculated theoretically, PIs, molecular weight, those can be done just based on the sequence. But any hands-on data you have, because as you know, proteins fold differently, and a theoretical PI and an observed PI can be different, will help us to maximize your timeline and your budget.
You said that one thing that sets you apart is you're offering bespoke services, because choosing a development partner or a CDMO can be quite overwhelming. So why should I, as this fictional CEO, choose KBI, or why should I go to another CDMO? Like, what are some decision parameters I should consider?
So if you're looking for an IgG1, a very vanilla antibody, you probably don't need the services of KBI. So KBI has a very high advanced degree rate. We are very good at complex molecules. Our experience in bispecifics, conjugated molecules, the list goes on, but we're going to be more expensive than the Lonzas, the Thermos of the world. So if you just need an antibody produced, that's going to be your better path. If you need something that's going to have some nuance to it, that requires more thought, more experimentation, as I said, bespoke services, that's where KBI is kind of fitting in that niche market. We're not going to be able to compete with the huge whooshies of the world, but we are going to be able to compete with the experience that you would need to make your molecule that's complicated successful. We have a number of clients that request RFPs from KBI and then comment back that it's more expensive and they go to a cheaper CDMO. And then we get a number of those back, right? And so now they have sacrificed some of their money and had a failure at a more platform CDMO. And they've sacrificed part of their timeline because they had to take 6 months, 7 months to figure that out. So I think for that theoretical CEO, knowing how complicated your molecule is and do you need the nuance that we offer. Our batch success rate for fiscal year 24 was 93%. And we don't do engineering runs. So we put in front the cell line, analytical, and process development engine to make sure that by the time we're making it at the scale of 2,000 liters, you're actually going to get a product successfully.
Let's talk about the analytical puzzle, because, there's a lot of things going on. Yes, as we've highlighted, we have the antibody, the linker, the payload. We need all kinds of different analytical methods. So tell us, what is your approach? How do you develop methods to characterize each component? Because at the end of the day, you need to have a sophisticated process control that you're getting actually that very ADC you want to develop. Before we continue, Biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, "David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs." Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode.
Yeah, I mean, there's a number of compendial methods that are standard for any antibody. We also have product quality methods. Your classic SEC, we need to understand the HMW. You want to keep that aggregate low. That's not specific to ADCs or non-ADCs. When we get to specifics for ADCs, it involves characterization of the free payload, the free linker, and understanding the charge heterogeneity. That's usually where we see the most nuance required. Both with cytotoxic and non-cytotoxic, there seems to be more of a development challenge for the ICE or IEX methods that we're running. We see shifting profiles, we see non-consistency, and getting the separation, for example, in ICIF with the right blend of pharmalytes and the right blend of handling is critical to get us from the development stage to the qualification stage where we're actually generating GMP data, and then to the validation stage where we can execute this across multiple sites, multiple handling. It's very robust. That's the biggest challenge I've seen. The free payload tends to be a pretty straightforward reverse phase method where we're just titrating against the standard curve. It's the charge heterogeneity that keeps me up at night.
Okay. And what are the things you can do even upfront, like before we even would come to KBI or another development partner? Are there some strategic choices companies should make to avoid, for instance, these headaches? I mean, I know there's a lot of complexity, but are there maybe some decisions, some early decisions that simplify or reduce the complexity?
I think a lot of it is understanding your linker attachment sites. So the most successful conjugated materials we've made are things where it's very well known and understood, both the conjugation sites and then any of the post-translational modifications that may influence that. So we have dedicated mass spec team that's able to, we can produce forcibly degraded material. They can understand how it's going to degrade and understand that at the molecular level. These sites are prone to this. And then we can actually monitor that, for example, oxidation or any other modifications that we present through detailed peptide mapping. We can identify those individual peaks, understand when we see a peak grow in, what it means is happening. Peptide mapping allows us to do that in a more streamlined way once we've identified what each peak is. So up front, there's a little more development to say when this gets oxidized, this shoulder or this peak forms, but then we can monitor in a very cost-effective way over the duration of the product.
Had you mentioned that the linker is very important, and I imagine also the conjugation method. So can you tell us what is the state of the art today? Because, I mean, a lot of thing has happened in the last few decades in ADC.
I mean, there are a number of ways to conjugate things. Most of it is thioethers, disulfide bonds, and peptides. They're very successful. Having specific attachment sites, like I said before, is also critical. Not having it be a random attachment. That was the early ADCs where we were hoping for the best. there was hopefully a certain amount of payload attached. The actual conjugation decisions is a little bit out of my expertise. I'm usually dealing with the actual molecule that now has it attached and how we can understand that. How they make those decisions obviously also has a component of how efficacious it is. So we want to make sure that none of these sites are blocked that would need to bind for that to be effective. But we continue to refine as we learn. The ADC market is going exponential. There are 12 approved ADCs, but we're aware, and the market's aware, of hundreds of ADCs that are in development. Now, a lot of those won't make it, but some of these platforms can be applied multiple times. So once we have one with a certain client, that client will have a high rate of success.
Now, let's talk about the formulation, because that's also quite a challenge. An antibody now is quite well managed, but probably more complex modalities. How does that play out for an ADC? What are the challenges, and what approaches have you developed as a team to tackle formulation challenges?
Absolutely. That is definitely a component. We need to develop a formulation for the drug substance intermediate. That would be the pre-conjugation. And that formulation can't inhibit the actual conjugation reaction. So we need to be careful. We can't have any surfactants present. We can't have any small molecules. That would be an issue. So our classic approach at KBI is to have a very simple formulation that is stable to freeze thaw. So we have to remember that buffers and some excipients can actually have a shift during freeze thaw. So we don't want to damage the biologic while it's actually undergoing that process, the freeze and the thaw, for part of this drug substance intermediate to drug substance production. Once we actually have it conjugated, then it's more typical for us to have a small amount of surfactant to reduce stress and agitation and freeze thaw, as well as to add different molecules that will add to the stability. The gold standard for all biologics is 24 months at 2 to 8 degrees stability. So adding different components, different excipients to make sure that we can get that stability is the goal. But we have to be very cautious at the DSI step as opposed to the DS.
And for those who are not familiar with ADC, so the DS formulation is important because certain companies are not able to produce the antibody and then do the conjugation at the same site. So there's some shipment involved, right?
Exactly. So it's classic for non-cytotoxic drugs at KBI, as an example. We are able to produce the DSI, and then we can conjugate it to produce the DS. Then it's typically shipped to a fill finish facility, and it is made into the DP, which is typically a filtration and then filling operation. For cytotoxic, currently KBI does not offer that conjugation. So we generate the DSI and then it's shipped off to another site, which does that conjugation reaction, and then shipped to a different site, depending on the vendor, for DP production. But understanding the less handling you have to do of that material once it's conjugated, the better. Obviously, every operation increases the risk of damaging the payload, damaging the product, and also risk to employees. So we try to have a pH that we think is acceptable with as little amount of excipients that would inhibit no surfactants for the DSI, such that then they can spike in the additional things once it's conjugated. And then the DP is just a simple filter fill finish.
Oh, okay, got you. So you have a formulation for your DSI, you do the conjugation, and then you add whatever is needed to stabilize your molecule, right? Correct?
Exactly.
Okay, makes sense.
So the DSI and the DS are typically frozen formulations, so that needs to be taken into consideration. The DP is typically a 2 to 8 degree formulation. That's the most successful financially for companies when they're shipping to clinics. Everyone has a fridge. When you start getting into different temperatures, minus 20, minus 50, minus 75, you have concerns with, do the clinical site have those freezers, and do they have to supply those to each clinical site?
And to what extent are the DS and the DP formulations for an ADC different from a standard map, or are they quite similar?
They are quite similar. It's typical for us to have a surfactant present. As I said, it reduces agitation as well as freeze-thaw impact. Depending on the molecule, we might need to have other stabilizers like arginine present to reduce aggregation. The pH is obviously critical. Different buffers can be more or less successful. That's generally determined empirically. Obviously, we have targets based on the pHs that we're looking at. And that's going to be a function of the molecule's PI. So seeing whether or not there's impact of the linker as well as the payload on the PI is important.
That wraps up part 1 of our conversation with Amanda Hoertz. We've explored her remarkable journey and the exciting world of ADC development. If you have process development or manufacturing questions, book a free call with me at Brühlmann Consulting/call, and I'm happy to help you get started. And please leave us a review on Apple Podcasts or whatever platform you found us on. It helps other biotech scientists like you discover the show. I thank you so much already, and I love hearing from you. So thank you very much for tuning in today. Stay tuned for part 2, where we'll dive into the analytical complexities of ADCs. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.